My pleasure to introduce Dr. Maureen Donlin. And Dr. Donlin is a research professor in the Department of Biochemistry and Molecular Biology at the St. Louis University, as well as director of the Master's Program in Bioinformatics and Computational Biology and director of the Research Computing in the Office of the VP for Research. And very affectionately, I can say that she works on Cryptococcus neoformans, and so that's very exciting and what my lab does as well. And so we're going to hear a bit about her work today. And so please join me in welcoming Dr. Donlin, and we'll go ahead and get started. I'll turn it over to you.
Okay. Thank you, Jennifer. And thanks for the opportunity to present to this group of new mycologists starting out. Let me go ahead and share my screen. Make sure I have the right spot. Okay, as Jennifer mentioned, I'm a research professor in the Department of Biochemistry. What I'm going to hopefully tell you a little bit about today is sort of anti-fungal drug discovery as it's done or can be done in an academic lab, sort of motivation for why you might want to do that, the current state of available antifungals, and some of how that can complement your own research. So I'm going to break up my talk. I'm going to tell you a little bit about who I am and my path to fungal research. Maybe it'll be inspirational. Maybe it'll be a what not to do in research. As far as my path goes, it's a little unusual. Talk about drug discovery in an academic lab, and then 2 example projects that I've been involved in on Cryptococcus neoformans. So in a map of the US, I'll tell you a little bit about my journey. So I grew up in a tiny little town in the middle of South Dakota, which is straight south of Saskatchewan, if you're more familiar with the provinces of Canada. Went to school in a slightly larger town in west central Minnesota at the University of Minnesota at Morris. It's a branch campus of the University of Minnesota. And it was here that I really kind of fell in love with doing science and thinking about doing research as a path for me forward. I kind of went thinking I would do, pre-med like so many students. And then I fell in love with chemistry because of some really talented instructors in the chemistry department. Then moved on to Pennsylvania, to the Pennsylvania State University, otherwise known as Penn State, where I worked on T7 DNA polymerase and got my PhD in molecular biology in 91. My husband and I had met at undergrad, went to graduate school together. We moved across the country to, why is this not going forward? There we go. To San Francisco, where I did my postdoc, completely changing fields in plant biology at University of California at Berkeley. Spent 3 years there studying the mutator transposon in ZMAs. And then we were ready to try and solve the 2-body problem of finding a position at least in the same town. He took a position at St. Louis University when we moved to St. Louis. I actually spent the first 3 years there in the Department of Chemistry at Washington University in St. Louis, where this is where I actually started to get into a more computational role. My role there was actually in curriculum development. In the chemistry department, they wanted to develop new laboratory experiments for the general chemistry students that involved a more biological focus, since the vast majority of the chemistry majors were pre-med majors. And this was 1994, which is when the World Wide Web really started to take off, although you could still count the number of websites at this time. And they said, and I don't remember if the PI on the grant or if I came up with it, but we thought we should do some tutorials online that supplemented what we were doing in the lab. And this is when I started to learn some computer programming, or at least scripting. I learned HTML, some backend programming, some C++. When this position ended, I actually moved to St. Louis University, where I've been ever since. So since 1997. The first 3 years I was at SLU, I was in the Department of Public Health, or actually the School of Public Health. And this group, the group that I worked in there did tailored health promotion programming. So it's sort of database driven health promotion, where if someone wants to change their behavior, say quit smoking, the approach was to have them fill out a questionnaire of the motivation for this change in behavior. Like, you know, okay, you want to quit smoking. Why do you want to quit? What are your triggers for smoking? That sort of thing. And then you would develop material to share with them that was tailored to the answers that they gave to this questionnaire. And they took on a project or two that didn't have the funds for a true backend developer. And so I ended up learning how to do database programming. And around 2000, the chairs of biochemistry, where I'm at now, and molecular and microbiology and the School of Medicine at SLU wanted to hire someone to provide bioinformatics support, and knew they really couldn't afford to hire someone who was actually trained in that, because it was relatively uncommon skill set back then. And so they approached my husband and said, given your wife's background in science and now computers, she should be able to do this. So I, with some trepidation, jumped in with both feet and said, sure, I'll give it a shot. So I spent a summer learning bioinformatics programming, which at that time was done in Perl. It's now been supplanted by Python and R. And if I had to make one really strong recommendation as you're starting out, if you have any inclination towards doing this type of analysis and find computer programming at all of interest, I would learn Python and learn R. It will serve you well in a lot of different ways as more and more science and experiments are done in a high throughput fashion. So sort of scientifically, I mean, these are some of the things that I did, but while the first 15 years I've been in this department, I did a lot of bioinformatics and actually biostatistical analyses for projects in immunology and cancer and in viral genetics. But this is also when I started my foray into working on Cryptococcus neoformans, because Jennifer Lodge was on the faculty in the Department of Biochemistry at this time. She's since moved on to WashU and is now at Duke. But when I came on board to the department, she reached out immediately and said, hey, I have a project and I really need some bioinformatics support. So she wanted to identify cell wall related proteins in Cryptococcus neoformans that they could target for gene deletions. Now, in 2001, we didn't have a complete genome for Cryptococcus neoformans. There was a genome published for Saccharomyces cerevisiae, and then we had a bunch of what were called ESTs, sequence tag. I'm blanking on what E stands for in ESTs. But anyway, we had some genome sequence in fragments for Cryptococcus neoformans, but we had the complete genome for Saccharomyces cerevisiae, or at least a genome, a draft genome. So what the idea was that she would identify proteins related to cell wall function in Saccharomyces, and then using BLAST and various computational methods, identify the homologous sequences in this compilation of genomic sequence we had for Cryptococcus. They would design primers and then do the knockouts. And so that was the beginning of my collaborations with Jenny, and I have been collaborating with her ever since on various projects. So in the time since then, I've done work on the sequence annotation for Cryptococcus neoformans. I've did a lot in the annotation of, that was for the serotype D. I did a fair amount with the serotype A, continue to do some of that with serotype A. And we've published, I think now 12 papers together and had a couple grants together. Later on, we had a new department chair in biochemistry who pushed me to get my own funding. And so this is when I started thinking about project work that I could do in Cryptococcus that was independent of Jenny. And we had at SLU a group of former Pfizer employees who had been involved in various drug discovery work at Pfizer, who had come to SLU to kind of do similar work. And so there was a cadre of people here with a lot of experience in drug discovery. And I thought, well, exploring some of the chemical libraries that were available to see if they inhibit Cryptococcus neoformans might be a way I could kind of chart my own path into independent research funding. So that's how I got started. So a little bit about the state of express sequence tags. Thank you. Just blanking on the name. You don't hear about those anymore since that's no longer a technology that's needed. So a little bit about the state of what's currently approved for antifungals. So these are for treating systemic infections. Basically, you have amphotericin B that's administered in combination with flucytosine. It targets membranes or agasterol membranes primarily. It disrupts the membranes. It's pretty broad spectrum. It's fungicidal. It's quite effective. It's also, unfortunately, very toxic and has to be administered IV. So it's not the greatest drug in terms of the actual treatment modality. There are the azoles, of which fluconazole is one of those. These target agasterol synthesis. Specifically, they're only fungistatic, so you get a lot of resistant strains to fluconazole and other azoles. They cannot be used in combination with amphotericin B because they're actually antagonistic to each other within a clinical setting. The newer, somewhat more newer compounds are the echinocandins, which come from the natural products from another fungi, and then they've been modified with various lipid moieties, and this is caspofungin shown here, which has a, I think it's a myristal tag, that target them to the cell membrane, and they inhibit beta-1,3 glucan synthase, which is synthesized as an essential part of the cell wall of fungi, and beta-1 glucan synthase is an essential protein. They unfortunately work against kind of a limited number of species. However, they are fungicidal. So, and they're pretty well tolerated because they target something that's quite fungal specific relative to human cells. More recently, a different beta-1,3 glucan synthase inhibitor has been approved for treatment of candidiasis, and that's Ibrexa fungerp. It's a triterpenoid. It's also fungicidal. I don't know how much in terms of how effective it is against vulvovaginal candidiasis, but it has been approved by the FDA. And then the latest drug in terms of a limited approval by the FDA is Fosmanogepix. It was known as APX001. It's a completely novel inhibitor in terms of its target, which is in the GPI anchoring pathway, inositol acylation, or GWT1. It's quite broad spectrum. It's been fast-tracked for testing because it appears to have some efficacy against Candida auris, which has shown up as a new fungal infection that's resistant to a lot of different available antifungals. It's also fungicidal. So that's it for things that have approval that are actually in the clinic. A lot of the trials that are happening now might be derivatives or new formulations. So I know there's clinical trials of different formulations of amphotericin B. There is a newer echinocandin resifungin, which is in a clinical trial as well. As far as new completely new drugs or new antifungals that are in clinical trial, or I thought would go into clinical trial, there's EvoFunPath, which is targeting dihydroorotate dehydrogenase. It's in a phase 2b trial for invasive fungal infections in patients that lack any other treatment options, and in a phase 3 for invasive aspergillosis. When a chemist collaborator, Marv Myers, and I wrote this review last year, there was this compound ATI 2307, which was developed by a Japanese firm, it's an acrylamidine that inhibits the mitochondrial respiratory complexes 3 and 4 in yeast mitochondria, was recently purchased by the company Pili Therapeutics. And at the time, it looked like it was headed for a phase 2 trial sometime this year or next. But now if you look at their website, it's not even listed in their pipeline. So I don't know what the status of this particular compound is. It's undergone at least a safety trial, a phase 1 trial, but that's, but I don't know where it stands. It doesn't show up in the clinical trials database. And so that's where we're at. It's a pretty sparse pipeline for the number of fungal infections that are out there. And obviously with, as we were just discussing, with the climate change, it's probably already happening that we'll see more fungal infections and opportunistic infections expand their range. So with that in mind, I want to talk a little bit about how the drug development process works and why you might want to consider doing some aspect of it in your own research lab. So if you think about in very broad terms what it takes to do drug development, obviously you need to start with some disease or condition that you want to treat. We're obviously interested in fungi, so we're thinking about invasive fungal infections. And within that disease or condition, you need to find, well, you don't need to. Ideally, you have a target. Either it's a specific protein or some biological process that you want to target with a new compound to treat. And once you've identified that and developed some sort of assay or method to test compounds, you identify a library of compounds to test, to screen. And you screen that compound library. You identify hits, so you identify sort of a cutoff of what becomes a hit. And then you find a medicinal chemist. Hopefully you've already found one by this point. They are your new best friend. And you're going to go through a kind of an iterative process of, okay, I have these hits. I'm going to test them in different ways. The medicinal chemist will go back and do their magic and come up with some new compounds for you to test. And you kind of go back and forth and around until you get things that are, you know, they inhibit really well, they're not too toxic, and they become quote unquote lead compounds. As part of this, you're also going to be evaluating the pharmacokinetic properties of these compounds. And so here you might want to work with a group that does what's called ADME, which is the absorption, distribution, metabolism, and excretion. And sometimes they add toxicity onto that, so that you're not just developing a compound that can inhibit your protein or process, but you're also developing compounds that have potential to actually work and make it into, you know, get absorbed into the body, that go to the tissue that you want it to, that aren't metabolized to something really toxic and so forth. And at the end of this process, you hopefully have one or two lead compounds, and then you would take it into an animal model. And at this point, you're probably going to work with a contract research organization to do the animal model testing, because there's sort of, the FDA gets very specific about how they want this animal model testing to be done. And if all of that works, you apply for what's called investigational new drug status, or IND. And once that's approved, you take it into clinical trials. This is not a short process, and it's not a cheap process. To take a completely novel chemical scaffold all the way to an FDA-approved therapeutic can take over a decade and cost up to $1 billion, which probably brings up the question, why on earth would I think I could get involved in that? And there's a couple reasons. One is these early steps, the sort of preclinical phase, is an area that really a lot of the big pharmaceutical companies have gotten out of. So the Pfizers and the Eli Lillys and Mercks of the world have kind of abdicated some of this space, particularly for treating infectious diseases, when you think about the drug is probably going to be used for a relatively short time, they want to be in the business of selling chronic drugs to treat chronic conditions so that they can make as much money as possible. Let's not put too fine a point on it. Drug companies are there to make money. So there's room in this space in terms of identifying new compounds that could be taken further down the line. And it can complement your other research areas. So if there's a particular, pathology or process that you're interested in, you could start a drug screening campaign. You might not be going all the way to a therapeutic, but you might identify compounds that could be used as probes, molecular probes, or tool compounds that could help you interrogate your system. The initial steps are identical. And this kind of work can be packaged up in ways that make it attractive to students, particularly undergrads or med students. And so if you have a lot of undergrads where they don't have the time to commit to a long project, but they still want to get involved in research, this kind of drug discovery work, if your assay is set up in a way that's relatively short, they can chunk through. I've had undergrads work on all my projects. They can chunk through that in a few hours a week and generate the data for a paper. So it has that advantage. At least at the NIH, there's funding mechanisms for funding this kind of work. STTR or SBIR, they're small business tech transfer or small business innovation research funds for doing that. I will plead ignorance of what the Canadian system has in terms of funding for these sorts of initiatives. So there might be some reasons why you would want to do that. If you decide that still sounds attractive enough, then what are the next steps? And probably it helps to understand what makes a good drug, because that sort of drives how you do the screening and why you do the various steps in that iterative process of optimizing that you do. So a good drug has obviously got a really high level of efficacy, and this is driven by how tightly it binds to your target, and then its potency or inhibitory activity against that active site. Ideally, the toxicity is low, and that's driven by the specificity for a single target versus off-target effects, and what metabolic byproducts are generated as it's processed, particularly by the liver. And then you want a relatively low dosage so that you don't have to administer too much of the drug. It also reduces the cost. And the ultimate dosage that it takes is driven by, again, how specific it is for the target protein, its metabolic half-life. So this is where all that ADME work comes into telling you something about that. And its bioavailability, whether it can reach, in the case of, say, something like cryptococcal meningitis, does it actually cross the blood-brain barrier? Does it get to where it needs to be in order to be an effective drug? So there are also there's different sort of drug candidate groups. Much of sort of what we do in drug discovery is built around small molecule drugs. So these are completely synthetic, typically less than 100 atoms, less than a kilodalton in size. It typically suggests that the chemistry is typically more tractable with these kind of small molecules versus, say, natural products, and typically have better oral availability, but that's not always the case. There's obviously a lot of natural products that are good drugs. The echinocandins are an example. Many of our antibiotics are natural products. We'll be honest, the fungi are the best organic chemists on the planet. We can't even begin to touch what they're able to do in their systems. And there's actually a fairly large amount of work being done around sort of genetic or metabolic engineering to modify natural products using the fungal species themselves, as opposed to trying to do it in the laboratory. One of the reasons natural products sometimes are a better drug than, say, a small molecule is that intrinsically they're more complex. They have more rotatable bonds, which makes their synthesis more difficult, but probably also increases their specificity for a specific target. They tend to be larger. They're usually more than a kilodalton in size. And that may, I mean, so they potentially bind to more of the protein and bind to it with a tighter affinity. And then, of course, there's a whole range of biomolecules that are also used as therapeutics. So they're, peptides and monoclonal antibodies of various types. But I'm focused mostly on the small molecules for the rest of this talk, as far as where you can go with developing or getting into this field for your own research. Okay, so all right, you think, well, okay, maybe this is something I want to do. So where do I start? And it was kind of hard to figure out sort of how to start, but we'll just start with what libraries you can screen, what are out there, as far as a starting point for screening, looking for new chemical scaffolds or new drugs to target your organism of interest. So there's a library of FDA approved drugs. It's 892 approved compounds. So these have been all the way through FDA approval. And they include a lot of, anti-diabetic, anti-seizure, and so forth. There is, and have been groups that have screened these approved drugs for looking for drugs that could treat different conditions. And so there were a couple of them that came out in the cryptococcal field, sertraline, which is an psychotic drug, and tamoxifen, which is an anti-cancer drug. And both showed that they had pretty decent inhibitory activity against Cryptococcus neoformans. Both ultimately ended up in a clinical trial to test to see if you add these to the existing standard of care, does it improve the outcomes? And unfortunately, in both cases, the answer was no, it didn't. And in the case of sertraline, there were too many adverse events around the fact that it's an anti-seizure drug or an antipsychotic, I forget which it is, they had to terminate that study early. So that's called repurposing. There's advantages to repurposing, not the least of which is that all the ADME's been done, all the toxicity's been done. It's a drug good enough to get into a clinical trial, so it has it can shorten the time to use, but the downside is it's been developed for a different use, and so it's unlikely. Really, what you're looking at is probably a side effect of it, or a side activity, and so in the end, whether it would ever be useful without some additional modifications is kind of probably a long shot. If you don't know what your target is, and even if you do know your target, there is a library known as the ICCB known bioactives library. So these aren't approved drugs, but they are compounds with known targets, at least known human targets. I was part of a study in Jenny's lab where we identified an inhibitor of an agonist of cannabinoid receptors in humans that also targets chitosan synthesis via the GPR4 protein, which I'll describe a little bit more. You can also, if you have a sense of what the chemistry is that you're targeting, you can work with a medicinal chemist, and there are companies out there where you can say, I want drugs that maybe target agosterol biosynthesis or this enzyme in agosterol biosynthesis, and they can help you sort of customize a library to test. And then the final I shouldn't say the final, but another approach is actually doing in silico screening or virtual screening. And this means you have a known or predicted three-dimensional structure, and you do literally virtual screening, and you can do millions of compounds this way and then identify a subset to maybe go after in a physical screen. Unless you have been hiding under a rock for the last couple of years, you probably have heard about AlphaFold, which has made huge inroads into making predicting three-dimensional protein structures, and has made this whole process much more feasible. Because before, if you didn't have a structure already, getting to a functional three-dimensional or usable three-dimensional structure was way too much of a hurdle for most labs. With AlphaFold, this actually becomes more feasible. That's literally a whole lecture in and of itself. So I'll stop at that point and say, okay, you want to do some sort of physical screen of some library. So in my case, where I started was, well, I'm going to work on Cryptococcus neoformans. I'm willing to just start screening some libraries and see what I find. In the process of working with a chemist here at SLU, we found out that cyclopirox, which is actually it's an antifungal for topical fungal infections. It's been around for a couple decades. It's a relatively small molecule. It's an N-hydroxypyridone. It's fungicidal. There were papers that showed it had broad activity across multiple fungal species. Not much in terms of the chemistry had been done, and there were no derivatives that had been tested in Cryptococcus. So this was sort of the starting point that I had when I kind of jumped in. One of the compound classes that I studied in Cryptococcus neoformans. Okay, so as far as testing, screening a library, what you want to have is a testing funnel. So I'm going to show this in kind of a high level and then talk about the sort of iterative process. So either have some high throughput or virtual screen, and how fast you can do this and how cheaply you can do this will determine the scale at which you can screen compounds. So the faster, the more reproducible and the cheaper it is, the more compounds you can screen. And you need to define a cutoff. So you want to reduce the number of compounds that you're taking forward at every step to the minimum possible, because you want to keep this whole process moving along and keep it as cheap as possible. So your cutoff would be either some predicted affinity if you have a protein target, or an inhibition constant, or in my case, a minimal inhibitory concentration. And then once you've done a primary screen, you might have a secondary screen. In my case, I'm looking at clinical strains of Cryptococcus, but you might have maybe you have a few concentrations or a single concentration you test at here. And then maybe in the secondary screen, you say everything that's, inhibits or binds with a KD of less than 5 micromolar, I'll then do a more quantitative affinity measurement. And then you want to have, as part of your initial screen, you want to start doing cytotoxicity assays pretty early on, so that you're not just optimizing for inhibition, but you're also making sure you're not just developing something that's really toxic, because it's hard to synthesize your way out of toxicity once you get down in that path. So typically cytotoxicity assays are done in liver or kidney cell lines. Not always, but we did ours in liver cell lines. In my case, I was using a minimal inhibitory concentration measurement, and then you would measure the cellular cytotoxicities, or the concentration at which inhibits 50% of the cells. And from this, you're getting a selectivity index. So the higher this, the better. So you have a high, so you have a very low cytotoxicity. Sorry, I have this inverted. You want it actually the other way, because you want a high cellular cytotoxicity and a low MIC. I have this completely inverted, my bad. In any case, once you've sort of gone through this screen, then you might do a secondary or a specificity assay. And again, depends on what you're looking at. You might be looking at off-target proteins, different species, and so forth. And then ultimately, you would get down to a couple lead compounds and you do animal testing. But this part of the testing funnel is where it gets pretty iterative. So in the case of what I did with cyclopirox, my primary assay was pretty simple. It was just inhibition of Cryptococcus neoformans growth done in a 96 well plate, picked conditions that were reproducible. RPMI is kind of the standard for other fungi in doing MICs. Cryptococcus really does not like RPMI much, so we ended up using YNB pH7. And you could either measure by optical density or resizurin, which measures mitochondrial function. And then I'm just looking for at what concentration does it inhibit 20% of the cells relative to untreated. So in this particular assay, it was somewhere between 0.78 and 1.5 micromolar, or an average of 1.1. If you want to screen a whole lot of compounds, or you have, say, a biochemical assay, your ideal primary assay, you want to scale down to at least 384 well plates, if not 1536. Ideally, you're using robotics to do the liquid handling, and you have some sort of colorimetric or fluorescent readout from that. And then this is that iteration by structure activity relationship. So your chemist is designing the compounds, you do your primary screen, you do your cytotoxicity screen, you give them back the data, he or she then goes back, makes some new compounds, you continue to measure this, and so forth. So you cycle through this quite a bit to ultimately come up with compounds that you can then take on to do further characterization of. I also did competitive inhibition with fluconazole just to see that, with the assumption being that anything that we might develop, we would hopefully be able to do in a combination therapy that might improve the treatment regime. So we ended up with this small project having 41 N-hydroxypyridone derivatives to test. These were the MIC and CC50 ranges. So the selectivity index was from, I think I still did it wrong, 1.5 to 19. We saw similar inhibition of fluconazole-resistant strains of Cryptococcus neoformans and gaddii that I obtained from John Perfect's lab. And we counter-screened ultimately in a couple other fungal species, Candida albicans, Saccharomyces cerevisiae, some clinical strains of that. And then a couple Malassezia strains, which we obtained from a collaboration we were doing with a mycologist in the biology department. It turned out that Candida and Saccharomyces were inhibited at pretty similar levels, but the Malassezia species were not significantly inhibited. And this is what we learned from the SAR was, so if this is the sort of the structure of the N-hydroxypyridone, we were able to modify these R groups. So this is R1, R2, R3. So we could modify 4, 5, and 6. Here's cyclopirox, the starting point. If we moved the group from R6 to R5, if we didn't have a group here, it increased the MIC quite a bit. We could change that group at R6, and as long as we had that lipophilic linker, it was still a pretty decent inhibitor. We could remove the methyl group from R4 and still had a pretty decent inhibitor. So that's, it's a very limited SAR, but we'd hoped that it was enough to at least put into a grant proposal for R21 funding. Ideally, you would know your target, you'd know the mechanism of inhibition. In all honesty, cyclopirox is an approved drug without them knowing. They don't know what it actually inhibits. It's thought to inhibit metalloenzymes by chelating the metal ion while it's bound in the active site. So we thought, well, okay, we'll take cyclopirox and 2 of the best hits and just try to explore whether we can figure out what it's inhibiting, or at least some broad parameters around how it's inhibiting Cryptococcus neoformans growth. And so one of the questions we asked was, if we add exogenous metal ions, does that rescue inhibition? So we used we use the drugs or the compounds at a little about double their MIC. So we use cyclopirox at 2 micromolar and the 2 derivatives at 4 micromolar. And then we increased concentrations of iron, 2 sulfate, copper sulfate, or zinc sulfate, and asked, does it rescue the inhibition? So here's, with no exogenous metal, you can see the inhibition. And then as you increase the iron sulfate, even at 0.25 micromolar and 0.5 micromolar, it completely rescues inhibition by all 3 compounds. The copper sulfate rescued the inhibition with cyclopirox, but not as much of the 2 SLU derivatives, a little bit more for this one than this one. And the zinc sulfate really didn't rescue the inhibition at all. So it kind of points towards iron, but maybe the copper is doing something, or maybe these 2 derivatives target something slightly different, or target an enzyme slightly different than cyclopirox itself. So then we turned around and said, do chelators of these metals synergize with the compounds, with the N-hydroxypyridones? So if you've never done a synergy assay, I'm just going to quickly step through what that looks like. So in fungal or bacterial inhibition studies, a synergy assay is set up as a checkerboard where you have across almost all the cells the same concentration of cells. And then you have, in 2 different directions, you have dilutions, serial dilutions of 2 different compounds. So you have, here's one antibiotic going from right to left, increasing concentrations, and a second antibiotic, in this case, going from bottom to top, increasing concentrations. So here's the positive control. This is the cell growth with neither of them, with just one of the 2 drugs. So this one's at 0. You can see where this one inhibits at about 4 micromolar. And on the bottom row is the concentration at which the second antibiotic, not micromolar, micrograms per mil, the second antibiotic inhibits at 32. And then when they're in combination, when there's both drugs available present, you can see that this one decreases from 32 down to 4, and this drug is decreased from 4 down to 1. So you can measure, basically you get out of this what's called a fractional inhibitory concentration, so the concentration of one drug in the presence of the other, and a combination of that is the fractional inhibitory concentration index. So this part's relatively straightforward to calculate. Figuring out how to do these serial dilutions in 2 directions took me probably half a day. I did finally figure it out, but it was always interesting to teach the new students how to do it, because it seems like it should be pretty straightforward. It turns out you cannot use a multi-channel pipette in both directions. There's one step where you have to use a single channel. But anyway, so once you have figured out your FIC index, you can determine whether it's synergistic, whether the 2 compounds work, and that's an FIC index of 0.5 or less than 0.5. If they're antagonistic, so sometimes if 2 compounds are targeting exactly the same protein, they can interfere with the binding of each other, and it actually takes more drug, a higher concentration of both of them under those situations. So antagonism is an FIC index of greater than 4, and the vast majority sort of end up as additive or indifferent to each other, and that's somewhere between 0.4 and 5, 0.5 and 4. So we did this with 5 different chelators. 3 were iron chelators, 2 were copper chelators. And what we saw with all 3 compounds was that in the presence of, they were synergistic with the iron chelators, the EDDHA, deferoprone, and deferoxamine, but were indifferent to the presence of copper chelators. So we're like, well, okay, it's tending to point towards iron-dependent enzymes or iron-related enzymes. That is surprisingly unhelpful because there's more than 100 of those in the Cryptococcus neoformans genome. So we're a little closer. We've got it down from 4,500 genes now to 100. And we have some evidence that these probably target more than one enzyme, in part because we did test whether these three compounds were antagonistic or synergistic with each other, and they are not antagonistic with each other, which would suggest they're not targeting exactly the same protein. And there's published attempts in Canada albicans where they try to develop resistant strains to cyclopirox, and these failed even after months of exposure, which suggests that either they're targeting something that's highly essential, or they're targeting more than one protein. We have in Cryptococcus neoformans a gene deletion library, which now covers about 4,000 of the 6,500 protein coding genes. We cherry picked a couple of iron dependent. So ferroxidase is CFO1, CFT1's a iron transporter. The knockouts of these we tested with cyclopirox, and they don't appear to be any more sensitive than wild type is to cyclopirox. So, I mean, like I said, there's over 100 of them. So that was kind of a long shot as to whether that would work. The last thing we tested, so iron-dependent enzymes are pretty overrepresented in oxidative phosphorylation in the electron transport chain in mitochondria. So we tested whether there was synergy between various inhibitors of the complexes of the respiratory electron transport chain, and also AOX1, which is an alternative oxidase found in fungi. And what we found was that all three compounds were synergistic with rotenone and potassium cyanide, which target complex 1 and 4. And then it's also synergistic with sham, which targets AOX1. The AOX1 strain was also not more sensitive. So we're kind of left with, well, we're pretty certain it's iron dependent, but we don't know which ones. In the grant that Marv and I put forth, which unfortunately was not funded, at least not yet, we put forth a strategy to try to identify the target. And so here, Marv proposed developing a bifunctional photoactivatable linker probe. So you attach on to your inhibitor a moiety that is photoactivatable, and then in this case it was an azide group. So you'd incubate this bifunctional compound with your cell lysate, hit it with some light at 360 nanometers, activates this so that it will covalently bind to whatever your inhibitor is bound to. So hopefully this is your target protein. Then you have it undergo copper catalyzed click chemistry with biotin to make it a biotin labeled complex that you can then affinity purify using streptavidin beads, do a trypsin digest, talk to your friend Jennifer about doing mass spec, and identify the target by mass spec. There's a lot of controls that you need to build into this, but that's basically the general idea. And then this was his scheme that we put in the grant. Said we still haven't gotten the funding for this. We're hopeful maybe in the next round to try and do this. Okay, and then the second project that I was involved in is more about finding a tool than really finding a compound that would go into a drug. And this was driven mostly by a postdoc in Jennifer Lodge's lab, Brian Maybrook. And here he was looking for inhibitors of chitosan biosynthesis. So he developed what I thought was a pretty complex primary screen, involves flow cytometry and a fluorescent label for chitosan called Cibacron. And he could demonstrate that when you knock out all of the chitin deacetylase genes, so you have no chitosan, here's the relative fluorescence level. And then he screened the bioactives library. So 480 compounds. He did this flow cytometry screen, identified 8 hits, validated in a dose response proliferation assay, and identified 2, validated these 8, found 2 more, confirmed those in a secondary biochemical measurement of chitosan, 2 different compounds which inhibit chitosan production in Cryptococcus. So what did these hits turn out to be? Well, one of them turned out to be BML 190, which the known target in humans is cannabinoid receptor 2, which is a G protein coupled receptor. So he looked at all the G protein coupled receptors. These are just the data showing that in wild type cells, here's increasing amounts of BML 190 at 10 micromolar, 5, 10, and 20 micromolar. It has reduced chitosan fluorescence relative to untreated. Here's the CDA 123 deletion, which essentially makes no chitosan. And then and this was the confirmation by biochemical assay. So then he looked at the G protein coupled receptors in Cryptococcus, and here's wild type reduction. Here's the GPR4 deletion. So there's no difference between when you treat with 10 and 20 micromolar relative to wild type. Here's the GPR2 deletion. You can see the decrease. GPR5, not a great, but there's some decrease. And then the CDA 123, which makes no chitosan. So he did a couple others, but he basically showed that BML 190 acts through GPR4, which is required for cyclic AMP production, which was not something that we knew a connection around. So even though we identified something that really only works at 10 or 20 micromolar, and that's not really a great hit, it did provide evidence that chitosan production is related to cyclic AMP production, which was not something that was known. And what we could do at this point, because the G protein coupled receptors, we could predict the structure for GPR4 and actually do potentially a virtual screen to identify other agonists, potential agonists, and see whether you could use this to interrogate the production of chitosan. So here's an example of a screen that ultimately identified a new, potentially a new molecular probe, but also sort of uncovered a connection about the production of chitosan and its interaction with a different pathway than we knew before. So I think that is all I have. There's just the acknowledgements of the people who worked on it in my lab and Jenny's lab, the people who are part of what is called the SLU Institute for Drug and Biotherapeutic Innovation, or IDBI. Marv Myers is the chemist in the chemistry department at WashU who I've worked with, and some other groups that I've worked with on other projects. But at this point, I'm happy to take any questions, and hope that you found that somewhat useful.